Okay, yeah, I can see the screen. If you are ready, please go ahead. Thank you.
Okay, it's just the presentation slide, right? Not the entire background.
Yeah, I think you
Okay.
are in the slideshow mode.
Okay, yeah, thank you. Hello, everybody. My name is Sobha Shanbag, and the paper that I chose to present is called the Annotation Efficient Cell Counting. And so before I start with the motivation for this paper, I thought I'll give you a quick introduction to the microscopy image. It's another modality of medical image analysis. And what it is usually a slice of a tissue that you will kind of look under the microscope on the right here. So why do we need to, what's the need for counting the number of cells in a microscopy image, right? So there are many I guess some of the needs is that it provides the information on the proliferation of the infectious cells. It also helps to identify the cancer biomarkers. It can quantify the T cells that invade the tumor. So it has a lot of applications in the lab setting. So these images can be used by deep learning to actually count the number of cells in these images, right? So what this paper, or what motivated this paper, is how to use deep learning models effectively to count the cells. Because we all know that deep learning models require a large amount of label data to train the deep learning model, and getting annotated data for these cell counting data sets is very tedious. Manual cell counting is not easy. It's literally a pathologist sitting and looking these images under microscope, and then counting the individual cells. So what this paper's motivation is, can we identify and annotate the important cell regions in a training image using active learning? So that is the motivation for this paper. So the paper actually does a very good job of looking at the existing active learning suggestive annotation models, and it gives you three main problem statements or research questions that it tries to address in the paper, right? So the first one is, can we label only the most informative regions of the whole image, hence reducing the burden of the pathologist? So they can look at only the most valuable section of the image that is that they can utilize for annotation. And then the second one is, can we identify and cluster the regions with similarities and use the suggestive samples from the different clusters for labeling so that the unlabeled data is well represented? And the third one is, can we train the model with both labeled and unlabeled data together in a semi-supervised fashion to capture the visual representations and select the most valuable unlabeled data for annotation? So these are the three main questions that this paper tries to address, or the problems that this paper tries to solve. So before I move on, I thought I'll give you an example of annotated cell image. So on the left is a cell of a human adipocyte cell. It's from one of the data sets that is used by this paper for counting. So on the left, it's a cell image, and on the right is what this annotated image looks like that this paper uses for training. So going into the proposal and contribution of this paper, the paper actually gives you a new active learning algorithm using an encoder and a decoder architecture. And the next three bullet point is, what is the novelty of this paper? Because what it does is, in their initial analysis, they looked at all the existing papers out there, and they tried to pick the deficiencies of these papers and then combine them into a new method or a new proposal, right? So they calculate the uncertainty on the models and also the uncertainty among the training epochs, right? So uncertainty actually represents the reliability of our inferences. So they're trying to see how reliable is the samples that were picked for annotations. So the epoch-wise uncertainty is actually a novelty for this paper. And then they also look at the diversity of the unlabeled data that they pick for clustering. And also they they also make sure that, or they tries to preserve the representativeness of the samples that they pick for annotation, and also rarity of the unlabeled data. So that is actually, I believe, a very good feature, because they are actually comparing the samples that they pick for annotation against the already existing label data to make sure that they pick samples that are not already labeled. So these three together is the heart of this paper. And then they also go into suggestive annotation. They only pick the important regions or patches of the images, and these patches are then recommended for annotation. So this will alleviate the annotation workload for the pathologist or the biologist who is trying to annotate these images. And then the label samples and the unlabeled samples are used in a semi-supervised way to train and fine-tune the cell counting model. So this is the main contributions of this paper. And the model uses active learning and multitask learning to generate the samples for expert annotation. So I thought I'll give a quick intro to both of these new terms, because it was new to me. I'm new to deep learning, so active learning and multitask learning is something I had to look into. And so what active learning is actually it reduces the burden of training on an unlabeled data. So it tries to identify the most interesting and the rare features and choose them for annotation. And the active learning also attempts to maximize the model's performance gain while annotating the fewest sample possible. And then going into multitask learning, all it is it's just using multiple tasks, multiple learning tasks are solved at the same time, while exploiting the commonalities between these different tasks and across these different tasks. And then finally, they use semi-supervised learning to exploit the unlabeled data to get better feature representation and to improve the algorithm learned on the annotated data. So going into the high-level approach for this for this paper, so pretty much these encounters are encoders and decoders. These are already trained on the labeled and the unlabeled data. I'll go into that a little bit later. However, the approach starts with feeding the unlabeled data for suggestive annotation into these already trained counters. And then these trained counters generate density maps. This is E is the number of epochs, and then N is the number of counters. So they generate E by N density maps. And they also generate the cluster labels of all these unlabeled data that is being fed into the counter, right? So they group the unlabeled data by deep clustering to guarantee the diversity of the data. So the encoders, decoder counters actually output these two outputs. And then these two outputs are then sent into uncertainty evaluation to pick the best possible samples for annotation. So they go with they evaluate the uncertainty a few different ways. They go with the model-wise and epoch-wise uncertainty to pick the most diverse samples, and then also pick the most representative and rare samples. And then these samples are sent to a human expert for annotation. And then these annotated samples are then fed back into the counters for fine tuning. So again, once they get the set of samples back, they train or fine tune the counters again on the entire labeled and unlabeled data set. And then this process kind of iterates over and over till one, if the model converges, good. If not, they have a certain allocated budget for the expert. So if they ran out of the budget, they cannot annotate the samples anymore. So that also stops the training. So that's how they go with the iterative approach in this training process. Going into the details. So they use a pre-trained VGG16 model for the feature encoder. The feature encoder actually generates the feature representations that can be used for suggestive labeling. So it spits out a bunch of features, and then these features are sent for feature enhancement module. So the feature enhancement module has three parts. It has a spatial attention mechanism, which pays attention to the important features spatially. And then they also have channel-wise attention mechanism, which assigns different weights to the feature maps for their two tasks, because they have these two tasks which is part of the multi-task learning. And then finally, they have these convolution layers to extract features at different scales. So once the features go through this feature enhancement module, then it is fed into the two different task branches. You have your feature decoder. Its actual job is to spit out the density maps or the counts for the input image. And then you have your classification module, which generates C clusters for the unlabeled data to be used later for suggestive annotation. So on the left is the actual heart of the counter. And then on the right, the image shows how they go about with their semi-supervised learning, right? So you have your labeled images and all the unlabeled images. So the labeled images go through regular training, and then they use the MSE loss to calculate the loss for the labeled images. And then for the unlabeled images, what they do is they send the original image through the counter, get prediction, and then they also send the augmentations of that unlabeled data to generate the count data. And then they find the cross entropy loss between these two, predictions. And both of these combined feed into the total loss, and then this total loss function is used to train these feature encoders and decoders. So I've included their loss function here just as a reference. The first half is the I believe the label data loss, which is the mean squared error. And the second one is the cross entropy loss for the augmented and unlabeled data set. And these lambda 1, lambda 2, and lambda 3 are weights. They already trained and set certain values of 0.1, 0.01, and 0.001 for these based on cross-validation that they ran previously. So going into the suggestive sample, the sample suggestion for annotation. So this is actually one of the main features of this paper. So the trained cell counter, which estimates the density map and the cluster label, and that information is used to capture or suggest the samples for annotation. And it's based on three different, features or methods. The first is the uncertainty score. So the uncertainty score, they use two methods to capture the score. One is the model-wise uncertainty. So they use bootstrapping strategy that trains n cell counters, each of which randomly select a subset of samples for suggestive annotation. And then they look at the disagreement between these counters and how they selected these different samples for suggestive annotation. And that is what the model-wise score is. And then they also calculate the epoch-wise score. And this is actually the novelty of the paper, I believe. The trained model is then saved every R epochs, and the epoch-wise uncertainty score over the model-wise uncertainty score is calculated to make sure that they are selecting the most representative samples. And then they also look at the diversity of the samples that they selected by clustering. The majority voting is used to assign cluster labels for the unlabeled samples. And the samples are sorted on their uncertainty score, and the top case samples are selected for clustering. And from these clustered samples, they pick the most representativeness and rarity. So representativeness just means that they're picking the unlabeled data so that it improves the model generalization. And then also the rarity is they're comparing with the existing label data to make sure that to avoid labeling redundancy. So they are picking unique and unseen labels for suggestive annotation. So the experiments that they completed are based on 4 publicly available cell counting data sets. These seems to be the standard used by a lot of cell counters out there. And They are like the benchmarking data set. So the first one is a synthetic cell data set. The second one is a bone marrow data set, and then human adipocyte and a dupling cell counting data set. And the way they ran the experiment is they cropped the training images into 4 non-overlapping patches and padded them to the original size. And then they applied data augmentation to the unlabeled images to avoid overfitting. And then training samples for 4 data sets were generated, 1,000, 300, 1,000, and 2,000 respectively. And they used mean absolute error as an evaluation metric for the model comparison between these state-of-the-art models. That's how they conducted the experiments. They did not go into details on the hardware that they used or any other details. So going into the training, they first trained the counter in a semi-supervised way using both labeled and unlabeled data. And then they moved on to the annotation round. They used the 3 trained cell counters for suggestive annotation. And then the unlabeled data is then divided into 5 clusters. And then they select the 10 most uncertain representative rare samples from each of these clusters, and 10% of the total training samples are suggested for annotation based on the suggestive sampling algorithms that they use. And then the expert annotates these selected samples, and then they are fed back into the encoder decoder for fine tuning. So going into the results, they compared their results with 4 public data sets that I mentioned earlier, and they also compared their results with what they call 2 state-of-the-art cell counters and 1 state-of-the-art suggestive labeling counter. So these 2 lines, the red dotted line and the green dotted line, is actually the measurement of the cell counter performance, right? There are 2 parts of this paper. One is the cell counter and one is the suggestive sampling, right? So they measured both. So if you look at it closely, you can see that the cell counter itself did not perform that well. It was comparative with the existing state-of-the-art methods. However, if you look at the suggestive labeling performance, that is where this paper kind of shines. They were able to demonstrate that their proposed method was very effective in suggestive annotation of very low label data. So if you look at the 10% label data, the red line indicates the paper. And then the blue line is an existing state-of-the-art, suggestive annotation method out there called SORTA. And then the very top line is the random selection. They just pick a sample randomly for suggestive annotation. You can see that the paper actually consistently performs better for suggestive annotation task compared with the one method that they tested against. And also they also included some the predicted density maps for some of the image data sets out there. So you can see the VGG and the corresponding density maps and the MBM, the adipocyte, and the final, I forgot the name, but the final image data set. So they also conducted a ablation study, saw the different parts of their method. I guess they looked at the epoch uncertainty, which I believe is a novelty, and then your active learning and your semi-supervised learning. So they conducted without each of these and measured the MAE for against the 4 different data sets out there. And they were able to show that combining all of these 3 methods actually contributed towards an accurate counting result and a better suggestive annotation result. So the 3 main works that they kind of compare against are the ones that are listed here. The first 2 are the cell counters without suggestive annotation. And then the last one here is the suggestive annotation method called SORTA. So their comparison is not very significant. They're just going against these 3 different papers for comparison. So going into the conclusion and the strengths of the paper, They do present a very good explanation of the shortcomings of the existing cell counting models and the suggestive annotation models. And they clearly list the 3 challenges that this paper is based on. And the novelty lies in the use of many different methods like active learning, semi-supervised learning, and multitask learning to implement the solution. And they also consider the model uncertainty and the training epoch uncertainty of the unlabeled samples, and use clustering to capture the rarity and the representativeness. The testing results does show the proposed suggestive annotation algorithm is effective for the cell counting task with limited training samples. And the paper is well written, it's easy to understand, and it gives a very detailed technical explanation of their approach. And the code is also freely available for you to run. So going into the weakness of the paper, the authors include testing comparison with state-of-the-art cell counting methods. However, it's lacking in the details on how they conducted these tests and the specifics of testing and analysis of the results. The testing is just confined to, I would say, 3 paragraphs, that's less than that. And they also don't go into detail on how they choose these network parameters like the cell counters, because they kind of defaulted to 3. And then the clustering details and the selection of the k-means clustering, which actually will define the rest of the selections or the rest of the clustering during the training of this method. So the improvement over the state-of-the-art cell counter is not significant because that is the heart of the method. You're counting the number of cells. They should also include additional cell counting methods in their testing comparison. And the model includes expert evaluation for suggestive labeling. However, they don't give any details on or examples on the suggestive labeling that was written by the expert, and then how it was fed back into the training or evaluation later. They just show just a picture of image of an expert, and that was the end of the detail on that. So based on weaknesses, I would give this paper a rating of 2. It's a very good paper. I would vote for accepting. The concept and the approach is well developed and documented in the paper. Technical details are explained well. The novelty lies in the combination of different methods for suggestive annotation. The testing did not feel complete and is lacking in detail. Then future work, improve the cell counter to increase effectiveness and outperform existing methods with significant margin. Use additional data sets and state-of-the-art methods for comparison. Include examples of suggested annotated samples and involvement of the expert annotation. That's all I could think of. And that kind of ends my presentation, Professor. So thank you all for listening, and questions.
Okay, yeah, thank you so much for the presentation. I think this topic is very interesting, not just for medical image processing. I think a lot of industries, when they do deploy, AI or machine learning model, they require to annotate some data. Active learning is frequently used because, we want to reduce the cost of labor, right?
Yeah.
You have a certain budget, right? The time and the cost. We want to label those informative samples that can improve the model performance, right? If you just label some easy sample, the model is already able to predict very accurately, then it's not going to help improve the model performance. So active learning is trying to finding those informative samples that can, maximally improve the model performance under a certain budget. And we do this kind of iterative, as this paper also present, right? Each time you can do maybe a certain percentage. And once you label the data using the human expert, and you can fine tune or retrain the model. And the next time you will also select maybe much better, more informative samples to constantly improve the model representation capability. Okay.
Yeah.
Yeah, so have any questions?
I have a question. So
Okay.
part of this has to do with counting cells in a picture, and it seems like the way that this is actualized is with a density map. And so I'm curious if there's ever a way that we go from like density map to actual cell count, or is everything just you learn from how close your density map is to the actual density map?
I think you learn from how close the density map is to the actual density map. Eventually you can represent these density maps as images, right? That's why initially I had to understand how does this equate to actual output, right? That's why I included that cell image and annotation. So you actually get these dots on that output image. And I believe you can use, compare these origin and the predicted and kind of measure how accurate the result was. That's my understanding. I hope that helps.
Yeah.
Yep. Yeah. Thank you.
Yeah, I can add maybe add some comments here. Yeah, so cell counting, it's just like in the typical computer vision domain. There's a task called crowd counting. So basically, you want to count how many people are there in an image in a crowd scene, right? So there's a very active field called crowd counting. So the simple and the straightforward way, well, let's just using an object detector, right? Like a YOLO or fast RCN, those object detector, right? And then we can tune this as a human detector, right? Then you have each person, do you have a bounding box, right? Then you just count how many bounding box are there. But the problem is that when there's a very dense crowd area, right, the human subject is very small, right? And also often they are highly occluded, right? Like in some of the like in a big gathering, right, in the far region, usually you only see like a partial of the head of the person, right? They are very small. So doing object detection is not going to be very accurate. And sometimes one bounding box may kind of crop out or cover like multiple persons, then you miss the detection or miss the count, right? So the density map is basically, if you look at the density map, it's kind of like a heat map, right? So for those, person region, or here, the cell region, you will generate like a heat map with a maximum value, okay? So when you can easily translate these heat map or density map into the actual number, right? You can using maybe some threshold, or you can just compute the value. You're doing some regression, right? Compute the value of the heat map, translate that to the actual number. Okay, so this way is more convenient and more robust as you're doing these like a bounding box type of object detection. So yeah, I think this density map right now has been widely used for this kind of crowd counting. So here, cell counting has the same issue, right? Because if you look at this figure here, a lot of cells, they are cluttered together, right? If you're using bounding box, that's going to be very difficult to separate those ones are very close to each other. Okay. Yeah, I hope this helps.
Yep.
Professor, I do have a question though. Here they selected the pre-trained VGG16 model, right? This in effect is actually a segmentation problem, isn't it? They're trying to segment out these individual cells. Is that true? And they didn't explain why they selected VGG16. Is that?
Yeah, I think the feature extraction part is not their like a main novelty, right? So essentially they just want to take any pre-trained model. Definitely you can they can use other type of network architecture like ResNet or EfficientNet that is pre-trained on ImageNet to help maybe improve the feature representation. But here I think they're just using the off-the-shelf VGG. Yeah, I think maybe they can explain a little bit more better, right? Why they selecting VGG is because, you know, it's just more friendly for their pipeline, or they just pick one, doesn't care, you know, which one to
Okay.
use. Yeah.
Thank you.
Yeah, here I do have one question. Here is the augmentation consistency loss. So can you explain like how do they do the augmentation?
They did mention certain, give me 1 second, I have to look it up. I don't recall it,
Yeah.
but they do apply, give me 1 second. Oh,
Yeah, maybe, yeah, you can
I
look at
can
the
look it up.
Yeah, so
They do apply rotations and many
yeah,
different augmentation methods,
yeah,
you know, and they actually multiply the
right.
data quite a bit.
Yeah,
So yeah,
yeah,
I can't
I
recall
didn't,
right away.
yeah. I also didn't recall the detail because I read this like very briefly. But from my previous experience, these augmentation-based consistent loss is very normal or commonly used. Maybe we can take image classification just to simplify the problem. Okay, let's just consider if we are doing image classification. And we are also using active learning, right? Trying to annotate the most informative images to for the next training iteration. So what you can do is take the unlabeled data, unlabeled image, right? You can do the different augmentation like cropping, rotation, mirroring, or maybe doing some color jittering, etc. Right? So you feed this different augmented version, but coming from the same image, right? Just like when we talk about self-supervised learning, right? We do the augmentation, right, to generate different positive, images as positive pairs. Here, similar idea, we generate augmentations from the same image, and we feed that into the same classifier, and trying to see, okay, how confident or how is the decision made by this classifier going to be consistent or not, right?
Okay.
If the classifier is very confident, or if this image is a simple image, right? No matter how you do the augmentation, the classifier will have very consistent result, right? This is maybe a cat image, right? No matter how you do the augmentation, rotation, cropping, whatever. If the classifier is confident that all these augmentations belong to the cat, right, with high confidence, then you can say that this image is kind of simple case, right? Because we try all these different augmentations, the classifier is able to make very accurate prediction. It's very consistent, right? But if it's not very consistent, let's say one type of augmentation, I predict this is a cat, but the other one, the classifier predict this is a dog with higher probability, then there's gonna be some conflict, right? So there's not much consistency here. Then maybe the classifier is not very confident or may not able to learn the representation of a cat category very well for this particular example. Then this could be considered a very difficult example, then can be selected for expert annotation. So that's the the consistent loss is being used to determine, right, how like the difficulty level of this input data. Okay.
Yeah, thank you for that explanation. Makes sense.
Yeah. Another thing I was just thought about this crowd counting, problem. It's not related to active learning, but actually is unsupervised learning. So which like a step further, right? So active learning, you still have to have some human labeling here. So I read one paper, they using a technique. So they are doing crowd counting. So the way they do this unsupervised learning is that, so they crop out a small region of the image. Okay, then they have another crop, which is basically an enlarged version of that bounding box. How do I annotate here? is there a way to for me to annotate here? Just
Yeah,
trying to see.
on the screen.
Yeah.
Yeah, I think, let me see. I don't know if I can, I'll have to give you.
Okay. Yeah, that's
independently.
fine. Never mind. So
Yeah, I don't
so
have
basically,
this.
as I was saying, right? So you have a region, small region, but you have an outer, like an outer box, a bounding box, which is have a cover a larger region. So the assumption is that these outer larger portions should have more count than their inner small region, right? So in this case, they can formulate a constraint, a self-supervised learning constraint. That is, the density map or the count of the outer larger region should be greater or equal than the inner small bounding box. So they are using this constraint as like the self-supervised learning signal to perform these like unsupervised crowd counting, which I think can be applied here because if the larger region, right, the bounding box, it should cover more cell counts as the inner, like small bounding
Okay.
box. So that's another maybe interesting, strategy for this cell counting as well. Okay.
Okay.
Any other questions? Okay, so if not, we can go to the next presentation. Thanks again, Sabah, and please don't forget to submit the slides so I can post on the Canvas. Okay, thank you.
Will do. Thank you.
Hello?
Can everyone see my screen?
Yeah.
Okay.
Okay.
I think I was muted. All
Okay.
right. Let me go back actually. So my name is Alejandro Zamora, and I'm presenting 3D transformer GAN for high quality PET reconstruction. So a bit of background on PET scans. They are positron emission tomography. It's a type of nuclear imaging technology. It is used to diagnose and prevent diseases. Its output is a type of 3D image. And then there's two types. There is a standard dose PET scan, and that's abbreviated SPET. This has a standard dosage of radiation exposure to the patient. It does provide a high accuracy image. And there's a low dose version of the PET scan, and it provides a lower dosage of radiation exposure to the patient, and it provides a more noisy data. And so the problem at hand is that SPAT imagery exposes the patient to harmful levels of radiation, specifically patients who need multiple scans, say they have an ongoing problem. And so this radiation, if used too often, can be very harmful to a patient. So in order to, and the LPET imagery has the opposite effect, where it's a lower level of radiation, but there's more noise and artifacts in the image. So it may lead to improper diagnosis. So a solution would be to generate SPET quality imagery from LPET scans. Therefore, you have less harmful exposure, but you're able to have enough data to make a good diagnosis. So the proposed method that the paper is implementing is a 3D transformer GAN. And so in this GAN network, they have a CNN encoder, which will be extracting low-level spatial features, a transformer network, which will extract the global semantic dependencies, and then it'll be decoded and produce a fake SPET imagery. And then the discriminator network would obtain the input sample. It would obtain the fake image that the generator produced, and a ground truth image of the SPET. And using the pairs of LPET and the ground truth SPET, and LPET and the fake generated SPET, that will determine whether these pairs are real or fake. The discriminator will determine that. And so an overview or kind of like a flow of how the architecture is going to work is that you'd get input of 128 by 128 and 128 pixel image. And you would feed that into an encoder. The encoder would then produce these like a feature, learned feature representation. And this learned feature representation would be channel size, depth, height, and width. And so this feature representation gets then flattened along its channel dimension, and these channel dimension will then be split into sequences, and these sequences will then be added with positional coding based on the 3 coordinate system, the depth, height, and width, and concatenated to these sequences, which would then be fed into a transformer. And they fit in these position codings to preserve some structural integrity of the features. Once these features are outputted from the transformer, they're reshaped into a learned feature representation similar to the beginning of the network. And then they are fed into a decoder, which are used to build or generate, recreate a fake SPET scan. And so there's 2 types of losses that are generated in this type of network. There's the loss between the generated SPET and the real SPET. And then there is a loss generated from detecting whether these fake SPET and an input sample are fake or true pairs. And that's done by a discriminator. So together, this this network will have the total loss computed by this equation down here. For a data set, there's 16 samples, and those were 8 samples of normal control, which are perfectly fine scans, no issues. And then there's 8 samples of typo, 8 samples of mild cognitive impairment. So an issue with the brain. The image sizes, I mentioned that before, so it's 3-dimension, 120 by 120 by 128. The data was obtained by this type of machine, this PET-MR machine made by Siemens. One thing I want to know about that is that this data set is not available anywhere. I've been trying to find it. It's not. It's not. It might be something proprietary that I've gotten. So, for the training, they use Atom Optimizer, and they use 150 epochs to train it. They started out with this 0.0002 learning rate for the first 100 epochs, and then they implement a learning rate decay for the remaining 50. The generator and the discriminator network do train alternatively. So the generator would be used to optimize the discriminator And then in the next iteration, it will do the opposite. It's going to be used to optimize the generator. But only the generator is used for reconstructing the PET image, LPET image. For the results, they scored pretty high across these different metrics. Here has some definitions for the metric: peak signal to noise ratio, structural similarity to the index, and this one's fairly common, the mean square error. And so they do score higher than all these other implementations, even if similar implementations like this 3D GAN. And they do also have a lower set of parameters, compared to the 3D and LA GANs. So that's probably attributed to them using a CNN to extract features in the beginning. Here's some visual results they posted in their paper, and so it's kind of hard to see. I think that's one of weaknesses I wrote, but it's some of these are kind of hard to look. But where the square is at is where they are showing kind of like a noticeable difference between the reconstructed LPET imagery. And so their proposed methods are on the right, and the ground truth is noted there. So the blue images on the bottom are a little bit easier to kind of visually comprehend. There's the more intensity of blue is the more error that was shown from that image. So all the way to the right is the proposed method, and it's obviously compared to other ones, it's the least blue written. So for the experiments, they since they're experimenting with or using a small data set, they did an experiment where they extracted 729 large patches of size 64 by 64 by 64 from the original image size. They did this so they could reduce GPU consumption, and also to help again with that low sample size. They used a leave one subject out cross validation. This is where they would use 15 of the 16 samples to train and save one to test, and they repeat it 16 times. So they would go through every single sample, pull one out, and test on the other ones, the ones that remained. Using the patches that they extracted and then to leave one subject out cross-validation experiment, they increased their training size from 15 samples of training to 10,135. So they did try to help with that small sample size. For the evaluations, this shows on the blue their baseline. So this is just a encoder CNN, decoder CNN. So no transformer, no none of their proposed method, no the descriptor. So and this scored the signal ratio metric at this score 23.74, and here's a view of a section of the brain that they produced using that LPAT, that quality. The second test they ran is just implementing a transformer to their encoder CNN and decoder CNN, and so that raised their performance up quite a bit. And by the way, the NC is the normal control, and then MCI is the cognitive, the set with issues, basically. And then finally, the proposed methods was scoring the highest, and so that that shows the most clearest produced imagery. So in conclusion, the proposed method reconstructs LPET images into SPEM images by using 3D transformer GAN architecture. It achieves state-of-the-art results in their data set when compared to other options. There's a low number of parameters compared to not all models, but some of the models that were shown. But they did have only 16 samples. And so they did mention there's a possibility for extending research to multi-model reconstruction model. So I wouldn't know how they would use that. They didn't really mention it, but maybe use something like Perceiver. So as for what I thought they had a really good explanation of the architecture. I think probably they went through every single section of the architecture and they detailed it. So it was an easy read in that aspect. The experiments showed like a good effort to try to combat their low sample size. So you know, using that leave one out subject out cross validation and using the patches, they were able to at least help with their small sample size. And then ultimately we're able to produce state-of-the-art results when compared to other models. So specifically ones using GAN network. For the weaknesses, there's quite a bit. So I think the first weaknesses was the data set just entirely. You know, 16 samples is just far too small to be an acceptance of any proposed model. You know that, you know, at least in the hundreds. But you know, being in a medical detection or medical computer vision task, you know, samples are hard to come across. So there was no link to the data set. I think I mentioned that before. So you know, it might be something proprietary or something that we wouldn't have access to. So there was no mention of how the data was annotated. Or is it by doctors? Was it by like a nurse? You know, it wasn't really explained at all. Like for example, who annotated data from what hospital? So there's other papers that detail that very well. I honestly don't think it's a novel enough idea. You know, again, networks exist, transformers exist. It just seems like they implemented this on a medical computer vision medical task. There's no you know, augmentations or maybe creating their own noise on some of the images, you know, to see if they can augmentate the data set. You know, that's one way they could have helped increase their sample size. And then there's no source code either. So some of the questions I had when reading the paper, maybe some of the parameters I didn't understand, I would go to the code to see if I could understand it, but they didn't provide that. There was no training time metrics, and by what I mean by that's you know, how long it took to train. You know, was it 3 days, 5 days, a month? You know, they didn't provide that at all. And then I wish there could have been more experiments. But I understand with what they had in terms of like the sample size, it'd have been kind of hard to do any more experiments with just 16 samples. But you know, maybe something else. You know, maybe experimenting with augmentations would have been nice. Ultimately, I rated this a 4. I just don't think there was enough, you know, sample size to really support them. You know, support the solution for this problem. I think if they would have used a bigger sample size, they would have fared a lot better. But it's just it could be overfitting. We don't even know. So, and then again, the proposed model wasn't novel enough, and I wish I would have more information about the data set. And then here's my references.
Okay, great. Yeah, thank you for the presentation. I have a few questions. So first of all, what is the motivation doing this from LPET to SPET? Why perform such task?
The motivation would be to reduce the exposure to the radiation that an SPET would cause to a patient. So if you can reduce the radiation, but still provide the same kind of quality of data, then a patient wouldn't have the risk of
Mhm.
you know, being exposed to that kind of radiation. So that would be the motivation to
I see.
to do that.
Yeah, so I guess the low dose will result in the degraded image quality, but maybe has the benefits for the patient, right? Because low dose of radiation exposure. Okay.
Yeah, exactly.
I also feel like obviously this is doing image reconstruction, right? They have the ground truth as PET image, then it's unnatural to using this kind of PSNR, SSIM, MSE to measure the quality of the reconstructed image. But from the practical use or actual application point of view, I don't think this is a very good measure, or at least to demonstrate the effectiveness of their method. I would expect experiment for some downstream task, right? After all, we are using this medical image to do some examination, right? Maybe to do some diagnosis of the disease, right? Say they are using this PET scan to diagnose maybe brain tumor, those things. So I was wondering, okay, their method is able to improve the PSNR as compared to like a existing method by maybe 0.7 or 0.8 dB in terms of PSNR. How does this improvement translate to improving the accuracy of the downstream like diagnosis accuracy, right? If I want to using the reconstruct as PET image to do the tumor segmentation, will this maybe improve 0.7 PSNR, resulting maybe like 2 or 3% increase of the segmentation accuracy, right? I think that's the key question here, right? So if they can demonstrate this, then I think their method maybe has a true value, right? Otherwise, if you improve the quality, okay, the doctor may see, okay, a little bit clear, but is it going to help like the following diagnosis is the question.
Yeah, I agree with that. There's no correlation of their metrics to the like endpoint result, which is a good diagnosis. And it's equivalent to just having like a really nice histogram filter, on an image. There's no real like,
right
purpose. If they would have had that in mind, maybe they would have done had a stronger, argument.
Yeah, I mean, for the natural image, you want to do some image reconstruction, right? It's very reasonable to using this kind of visual quality assessment, right? PSNR, SSIM, to see if the reconstruct image is going to be, visually pleasing, right? But for medical image, right, visually pleasing doesn't help, right? We want to see, how this is going to play a role for specific, the downstream diagnosis task. So I think this is maybe my biggest concern, right? So demonstrating these, well-known natural image-based, metric is okay, but not necessary to show the real value of their proposed method. But it maybe, these 0.8% improvement of PSNR doesn't help at all, right? For maybe tumor segmentation, or maybe even introduce some, artifacts, right? Because maybe reconstructed SPET maybe have some region which is maybe totally new or maybe doesn't exist. So that's also, possible. So I think that's maybe worth to further investigating. So I think if you guys are doing some similar project, doing this kind of image reconstruction using GAN or whatever other method, this is something you guys can think about, right? What is the right metric, to demonstrate, right, the reconstruct quality, does help. Okay.
Yeah.
Okay. Any other questions?
Yeah, so so this paper, does this, like, their method, are they essentially like trying to use the method that produces lower radiation, but their model kind of filters out the noise that the lower radiation PET scan produces?
Yeah, that's yeah, they're doing exactly that. They're trying to
Okay.
use the low radiation type of imagery, but upscale it so it's a higher resolution.
Right.
And
Okay.
doing
And
that, they yeah, go ahead.
okay. Gotcha. And so I'm not sure if PET scan, I don't know if MRI, if this is exclusive for MRI scans, but um like, you know, modalities, right? Like, I'm sure you can use different, like, flare, T1, T2. I don't know if you can do that with PET scans, but if you can, did the authors like ever provide a reason as to like why they used a certain modality for the PET scan, or?
They didn't really provide that. It's very odd of how they got the data set. Maybe they're not supposed to tell anybody, but they didn't really provide much information about it. They did, and it's kind of odd too because they did provide a lot of information about the machine itself that took the imagery. So what kind of algorithms they use to construct the the image,
Mhm.
but that like the machine algorithm. So,
Yep.
but and then also they mentioned some factors like how long the patient received the injection. There's like I guess there's like a liquid injection they take for the scan to take effect, and how
Right.
long they took the injection, from what time. So but that's about it. They didn't really
Yeah, it
explain why
looks
they only use this type of data. So
yeah, it looks like they use like a very kind of low quality sort of like imaging modality, because like, or maybe they tried with higher better resolution modalities, but I mean, given all the negatives that we've like kind of discussed with it, with their model, or not with their model, but with their paper, maybe it wasn't like, maybe it didn't produce significant enough results, as opposed to, I guess, using this lower quality
yeah,
imaging modality.
yeah, I agree. Some of these other implementations, like in this example, like say if we're not going to consider the metrics, right? We're just going to consider how well this looks compared to each other. It's hard to distinguish the ones, the proposed model
Yeah.
from like L1 GAN, 3D GAN. Like it might be just the image I took, right? But
Mhm.
I wouldn't be able to really differentiate at least the far four from each
Yeah, it
other.
looks like three of them are like essentially the same. That's the thing, I'm not able to,
Yeah.
tell apart. I don't
Eh,
know why they used PETs. Maybe they could have used MRI, but maybe they're just trying to optimize PET scans or something. I'm not sure.
Yeah, well, PET scans, from what I heard, is able to provide like a view of a working, like
Mhm.
your organism's working. Like it's like able to capture like
The
I don't know,
function.
like yeah, the function. That's
Oh,
the word I'm
okay.
looking for. And so that's one aspect that makes it very useful. But
Okay.
yeah, maybe that's one of the reasons. But they chose to use PET instead of MRI.
Yep. Gotcha. All right. Thank you.
Yeah.
Yeah. Okay. Any other questions? So I think this may be like a bad example, right, for the paper we reviewed, right? So there's a lot of limitations or weakness. So yeah, we can learn a lot from this kind of bad examples as well.
Yeah, I was wondering, like, the slideshow was short, I was like, I don't have much to go off of.
Yeah.
Yeah, so
Yeah, yeah, but I think overall this problem is interesting, right?
yes.
I get where they come from, right?
Yeah.
So yeah, like MRI, PET, different imaging modalities, they capture different functionalities, right? So but the nature of this PET scan requires this kind of if you want to do high quality image, maybe you have injecting like for a mono dose of radiation that may not be good for the patient, or they will take longer to imaging a scan. So they want to using kind of low dose PET scan, and hopefully using machine learning or like a image processing technique to enhance the image, right? So that can achieve the same, at least same quality as the as PET. But here, I think they are not doing a very good job to show the results they have really says that, right? Although, they show this kind of visual comparison, but as you guys mentioned, right, they look pretty much very close. It's really difficult to clearly see the advantage of the proposed method, right? Maybe small regions have some more contrast, but like I mentioned, right, how this improvement translate to, for the later diagnosis, I think that's the key. If they would carry out some experiment, right, say, okay, based on their reconstruct aspect image, they are able to maybe achieve better, tumor segmentation or detection accuracy. This may be more convincing, right? Their method is better. And also, I think the size of the data set is really small. I don't know if there's any public available data set they can use, or they didn't find any, so they just collect their data set themselves. But yeah, 16 is very small, and it is very difficult to justify the effectiveness of the method.
Yeah, well, maybe, a possibility they can have, use a larger data set, maybe off like a different organ or something, and then introduce noise and pre-train the model maybe,
Yeah,
and then try to implement this, have like a pre-trained weights from a large
yeah,
data set, maybe something related.
yeah.
Maybe they could have helped, but I really don't know. It could not really be their fault. Maybe they're just handed this data set and they were trying to make it
Yeah.
work, so.
Yeah, but I guess another alternative to do the experiment due to the lack of data set is we can do some simulation, right? So let's say we are able to find some aspect image, right? What we can do is we can do some, downsampling or maybe adding some noise, right? Just to simulate a downgrade or degraded, like the so-called LPET image, right? So in this case, they are able to, maybe fully verify the effectiveness of their method, maybe using a larger simulated data set. At least that, give the reader more confidence, right? Their method
Right.
may perform better. I think similarly for other data modality. So there's I think this task is similar to like image super resolution, basically just to enhance the resolution of a low quality or low resolution image. So like MRI, CT, scans can also be low resolution. During the reconstruction process, right? They in the case-based, they do the case-based reconstruction. So if you're using less samples to reconstruct data, the data can be low quality. Then you can using some of the computational imaging algorithm, right? Using machine learning to do some image enhancement. So actually we can simulate a lot of this kind of low resolution and high resolution image pairs, like for different modalities, CT, MRI, or PET. So I don't think the lack of data set is excuse. So this is another way trying to have more data set and with different image modalities, then they can apply their method, not just for the PET, maybe MRI as well, right? This will fully justify or maybe validate the generalization ability of their algorithm, right? So if they are able to outperform the other method consistently across multiple data sets with different imaging modalities, then I think that'll be more convincing to the readers. Okay.
Okay.
Okay, all right. So yeah, I think we can learn a lot, not from this paper. So some of the comments, especially when you are doing your project, right? When you present your project, your result, and you have to think carefully, right? How is your metric is going to convincing people, right? This is the right metric to evaluate the algorithm, and also the result is going to be very convincing to people. Okay. Okay, before we end, any other questions? Okay, how about your programming assignment 2? Do you guys already started?
Yeah, I have a question regarding that.
Okay,
So I just wanted to understand, like, for the sake of time, convenience, in your experience, would you say that like training the 3D neural network implementation is like takes longer time to do than a 2D?
yeah,
It does?
yeah, yeah,
Okay.
unfortunately, yes, yeah.
Okay. And then so if we were to do that, I think you mentioned there's like a Newton server, a UCF Newton.
Yeah, yeah. So I
So
think I have those account and the keys. If you want to use it, send me an email. I can send you the key, so you can using that key too. You don't
all
have
right.
you don't need to set up like a password. You're just using that key to verify login. Okay. And
Okay,
some of the
yeah.
instruction to using the Newton I've posted in the Canvas.
Yep.
Let me show you. So I believe I post here. Yeah. So yeah, the first one is like, how do you get started? Basically, if you have the key, you can use it to log in. So yeah, send me an email. I will send the key to you. Okay.
Okay, sure. Because I'm not sure if my laptop can handle that.
Yeah.
But
Yeah, I doubt your laptop will handle 3D network.
yeah.
One is for the memory issue, because
Mhm.
if you're using a larger like 3D unit, you may run into memory issue. So yeah, if there's a issue, maybe you should try 2D network, do this kind of slice by slice manner.
Okay. All right, sure. I'll think
Okay.
about that.
Okay.
Thank you.
Okay, no problem. Yeah. Yeah, so yeah, 3D networker, because of the 3D convolution, so it will eat up your memory quickly. So that's the issue. Yeah, but maybe if you are able to using the Newton server, using this kind of at least like 12 or 11 gig of memory, and you use a small batch size, maybe I don't know, may have to reduce to like 2 or 4. You can try that, see if it can fit. Okay. Okay, all right. Yeah, if you guys have any questions about the homework, let me know. And also on Tuesday, there's office hours. So you can go to the office hours on Tuesday, and we can discuss there as well. Okay, but I do suggest you guys to work on this as soon as possible, because you may run into different training issues. Okay. All right. Thank you, guys. We're gonna end here, and I'll talk to you next week. All right. Bye, everyone. Have a nice weekend.
All right. Thank you. You too.